So this is the last section for the chapter on plant reproduction, and it deals with how plants disperse on their own, but also ways that we can use. Hold on a sec. Trying to get my iPad set up. Ways that we can use plant reproduction for our own benefit as well. Fruits and seeds result from the sexual reproduction in flowering plants. Fruits are special structures that are adapted for dispersing seeds. A seed functions in the dispersal and propagation, which is making new plants. There are many plants that can propagate asexually as well as sexually. Now, the way that plants propagate asexually, I'll talk about at the end if I've got time. But quite often we use this, like I've used it before. Like a friend of mine gave me a piece of her mint plant, and you just put it in water and wait for the roots to grow. So you cut part of a plant off, have the roots grow, and then you can replant that as a new plant. I've also done that with Italian basil as well. You just cut a branch and then roots form and it forms a new plant. So that would be an asexual way to reproduce plants. But the dispersion is the distribution of the individuals. So like whether the seeds just fall off the tree and fall right next to the parent tree and develop there, or whether seeds are carried by the wind, water, or even animals. If the seeds or fruit is too close together, then the new seedlings have to compete for resources like water, food, and sunlight. One of the successes of seed plants is the development of structures that are adapted for dispersion. So, for example, when we looked at the seedless vascular plants like ferns, what else was there? Seedless vascular, the fern allies like whisk ferns, Silitophyta, and all that group. They are vascular plants, but they still rely on spores. So seeds provide an extra protection for the developing embryo. And then on top of that, you have the angiosperms, which produce fruits as well, which is even more protection. Fruits and seeds are dispersed by animals, wind, water, even forcible discharge, or at the very least, gravity. The smell, hold on. The smell of the fruit, as well as some fruit is brightly colored, like apples, oranges, lemons, things like that. And the flavor is going to attract animals. When the animals eat the fruit, the seeds pass through their digestive system. The fruits and seeds are going to be adapted for the type of dispersal they depend on. For example, in the bottom right corner, we see it's called a thistle. I think it's a thistle plant. And you can see it's got these really kind of light, wispy fringes on top of the seed. So this is an indication that it relies on wind to carry it. Is it orchids? Interesting. Looks like thistle. Doesn't look like an orchid. Anyway, so you can see that they can easily be carried by a light breeze. The coconut relies on water dispersal. So it makes sense because coconut trees generally grow near water. And so the coconut, obviously, we know that it has like this large air chamber in the center, which allows it to float. Some plants actually force the seeds out. This is it's called a tropical sandbox tree. It has fruits that throw the seeds up to 100 meters away. So that was he didn't film the fruit because the fruit is way up in the tree. But you saw from this, that's the fruit, and it explodes open and like flings the seeds away. Okay. Gymnosperms don't produce fruit, but the cones help to protect the seed and aid in dispersal. This is a pine seed. And the pine tree uses gravity, a little bit of wind because of the blade-shaped, what do you call it? I don't know, the thing that sticks out from the seed. The seed is right here, but it's got that wing, I guess that's what I was looking for. So this helps in dispersal, like that. So if you can imagine, if there's a strong wind, that it might blow them further away from the tree. Okay, now I'm actually going to skip past this because I don't think it's important. Okay, so fertilization is the beginning of the development of the fruit. The fruit is there to protect the seed, to aid in dispersal, but also to delay sprouting. So the seeds inside of an apple, for example, aren't, excuse me, aren't going to start growing from inside the fruit. All right, now we're going to look at the structure of seeds. So a seed is a plant embryo that is surrounded by a protective coat that's called the seed coat. Now, the structures of seeds are different among the major groups of seed plants. That's angiosperms, which includes monocot and dicot, and the gymnosperms. Now, unfortunately, because we're online, we can't do the lab on this because I have, if you want to do it on your own, that's one thing you can do. All you need is, I usually use kidney beans, the ones that look like this, because they're quite big and it's really easy to see this structure exactly on the inside. So if you want to do it, all you have to do is soak the bean overnight and then carefully split it open, because the bean is an example of a dicot. I don't think I have white beans downstairs. Maybe I'll try to cut one open. They're a bit smaller, but anyway, if you get a bean, generally you can cut it open, it like splits in half, and then on the inside you can see these structures. So for the dicot seed, most of the inside of the seed is the cotyledon, which is the seed leaf that is part of the nourishment for the embryo. The actual embryo is here. Okay, that's the actual plant embryo. Between the two cotyledons, there are three other parts. We have the radicle, which is the embryonic root. If you ever have seeds at home that you're trying to grow, like what we did before Christmas, I think, when you spray them with water, and generally you can see the radicle coming out from the seed. That's the first thing that comes out, is the embryonic root, because normally the root is the first thing to emerge so that it anchors the seedling into the soil. After that, you can see these other structures start to come up. Depending on if it's a dicot or a monocot, it might come up, it might come out curled like this because it's protecting the plumule. These are the embryonic leaves that are very delicate. If it's a monocot, it might just come straight up through the soil. But the radicle is the first thing that that comes out of the seed. Then you have the hypocotyl. Sorry, I'm gonna let Nat in. There we go. The hypocotyl is the stem between the cotyledons and the radicle. You can see that here. So it's just, it looks like the main part of the stem. And then you have the epicotyl, which is the stem above the cotyledons. So the hypocotyl and the epicotyl. Remember the prefix epi means upon, which basically means on top of. So you have cotyl, which comes from cotyledons. I'm not sure, hypo is usually less than, but anyway. So epi is on top of the hypocotyl. The epicotyl with the embryonic leaves is called the plumule. So I basically look at the epicotyl and the hypocotyl together as being the embryonic stem. You've got the radicle, which is the embryonic root, and the plumule, which are the embryonic leaves. Now, on the concave edge of the surface on both sides, you see a scar that marks where the seed was attached to the ovary wall. That's called the hilum. Now, for a monocot seed, you actually see the endosperm is separated from the embryo by the cotyledon. In a monocot seed, the cotyledon doesn't nourish the embryo directly like in a dicot, but it transfers nourishment from the endosperm to the embryo. So the seed takes up almost the whole grain. The wall is very thin and fused to the seed coat, which you see here. There is a single umbrella-shaped cotyledon pressed close to the endosperm. Don't know why they call it umbrella-shaped, but looks kind of like a liver. It's the shape of a liver. If you took the liver and stood it up on its side, that's what it looks like. Right, so the cotyledon doesn't store the nutrients, it just transfers it from the endosperm. So it's kind of like, if you remember human reproduction, if this was a uterus, you could think of the endosperm as being the mother or the mother's body. The embryo here is the fetus, and the cotyledon would be the placenta. If you remember the placenta, which is attached to the wall of the uterus and to the fetus through the umbilical cord, that's kind of like the human version of this. And then finally, we have a gymnosperm seed. A pine seed is usually the typical example of a gymnosperm. The embryo has 8 needle-like cotyledons, and it's interesting because the cotyledons kind of mimic the leaves of the tree, because pine trees have needles, not leaves. The embryo is surrounded by the tissue of the female gametophyte. And the haploid tissue of female gametophyte functions as a source of nutrients for the embryo, like the endosperm of the corn kernel. Now, the reason why seeds are such a huge advantage over spores is because it's an actual fertilized egg. It's an embryo. It is alive, but it will not germinate or sprout until the environmental conditions are right. This helps to secure the survival of that plant. It would be really stupid and useless if the seed germinates in the middle of the summer when there's no rain. Many seeds don't germinate even when conditions are ideal, which you guys experienced firsthand when you planted your seeds and nothing happened. So we call this state dormancy. It's kind of like when bears hibernate for the winter. They're not dead, but they're just at a state of reduced metabolism. Bears, when they hibernate, they are dormant, but they don't sleep continuously for 5 months. They're just like really lazy, I guess you'd say. We can also use dormant to refer to volcanoes. Volcanoes which do erupt periodically but are not erupting at the moment, they're considered dormant. Now, what conditions are needed for germination? There are a lot of them, but a few examples are the correct amount of water, oxygen, and temperature. Now, the specific conditions needed is going to vary, or is going to be different depending on what kind of plant you're talking about. I am currently trying to grow chilies that I started with grade 12 when they were in grade 11 doing this exact chapter. I bought habanero seeds, I bought jalapeno seeds, and I also just like from the market, I had Thai chilies that I wanted to grow because I really want to make hot sauce. And I'll take pictures after the class is over, but literally I'm only just starting to get chilies now. And that was when grade 12, who just finished, that's when they were in grade 11 last year. So it's taken over a year. And I was super excited because I finally have one habanero of all of the habanero plants that that they planted in the soil. All of the chili plants I have downstairs, except for one, it's all Thai chili. So yeah, it's really difficult to grow, but I'm super excited I have that one habanero. And you guys also tried for like a week or so to try to grow seeds. It's not as easy as just like throwing them in dirt and then they grow. Most mature seeds are dry, so the first thing that they require is water. This is one of the tricks that I use when I'm trying to grow plants, is that the same way that I had you guys do at school was to put them on a little bit of tissue paper, spray them down with water, and then cover them so that the environment stays moist. Generally after like a day, 2 days, maybe 3 days, you will see the radicle start coming out. And then that's when you usually transfer them to the soil. So the purpose of the water is to soften the seed coat and activate enzymes that will convert the starch that's in the cotyledons or the endosperm into simple sugars to feed the plant, because obviously it can't rely on photosynthesis when it's still in the dirt. As the embryo starts to grow, the softened seed coat cracks open, which then allows oxygen to reach the embryo. Seeds will also germinate only if they're exposed to temperatures within a certain range. A lot of small seeds need light for germination. So this prevents seeds that are buried too deep in the soil. They will not sprout. So this is a bean seed. There's the radicle, the first thing that comes out. And one of the cool things that we're going to study in the next chapter over the next few weeks is how the hell does the root know which direction is down? Like, no matter which orientation you plant the seed, the radicle is always going to come out and follow gravity downward. We'll talk about that in the next chapter. So this is now the primary root, which we talked about before. And now you see lateral roots starting to form. And this is the hypocotyl, epicotyl coming out curved. That's how you know it's a dicot. The reason why it comes out curved is to protect the really delicate leaves that you see here. Because if it came straight up, then those delicate leaves would probably get shredded. And these are the cotyledons here that are left over. And they will drop off when all of the nourishment inside the cotyledons is gone. So now that the leaves are out, you can see the apical meristem behind, there it is. You can see it growing there, and you can still see the cotyledons on there, even though it's already going through photosynthesis. Okay, that's annoying. Even though the leaves will have started photosynthesis already, the cotyledons are still there. Oh, now they finally dropped off. And you can see the lateral root system starting to grow and that apical meristem right at the tip. So this is where the plant grows, starting from the tip, and you've got lateral buds growing off the side. Okay. Oh, here we go. All right, so it's going to show us. There's day 8. So you can see the lateral root system starting to grow, but it's still, this is what's called a taproot because the primary root is the largest root. And the first lateral roots are going to grow out from the top down. More roots. Oh, there we go. Not nice, big, tall plant. Okay. Now, some seeds are going to germinate only when they're exposed to extreme conditions. For example, the black lotus seed only germinates after passing through the digestive system of animals. There's actually a very classic example of something called coevolution, where two organisms evolve together because of the relationship they have, and that is, I don't think it was this one, yeah. So there used to be a bird called the dodo bird, and there was a particular plant that would only germinate once it passed through the digestive system of the dodo bird. And then the dodo bird went extinct, which means that plant also went extinct. The reason why they have to pass through the digestive system, the acids in the digestive system break down the seed coat, and it comes out with natural fertilizer, of course, when the animal poops. That is a fertilizer. Apple seeds are another example. They have to be exposed to near freezing temperatures for several weeks before they sprout. This prevents the seeds from germinating in the fall, because if they germinate in the fall, that means that these tiny little delicate seedlings have to survive the entire winter. So usually you plant the apple seeds in the fall, then in the wintertime, they're exposed to that cold temperature, and then they sprout in the spring. So here is the process of germination between a monocot and a dicot. And basically, generally, I just run through it again. You've got this very delicate embryonic leaf system here, the plumule. So the hypocotyl is going to come up curved to break the surface of the dirt before the first leaves open up. Compared to a monocot, which has a very strong epicotyl, and it's got this protective layer around it called the coleoptile, and that protects it as it goes straight up. So imagine if you have these soft little leaves versus a kind of harder, straighter, pointier leaves that can come straight up through the soil. But for both of them, the first thing that comes out is the radicle. In beans, which is the dicot, the entire root system forms from the radicle. But for corn, most of the root system develops from the lower stem. You can see that here. That's the lower stem in the root systems develop there. As soon as the radicle breaks out of the seed coat, then the shoots begin to grow. As I said, the dicots come out hook-shaped to protect those embryonic leaves. As soon as the plumule unfolds, then photosynthesis can start. As you saw in the video, even when the embryonic leaves are out and going through photosynthesis, the cotyledons will still stay on until all of the nutrients are used up before they finally fall off. Now, the cotyledon of corn in a monocot stays underground, so it transfers nutrients from the endosperm to the growing embryo. The hypocotyl doesn't hook or elongate. It stays below ground. So you can see from the diagram, the plumule of the corn is protected by this coleoptile sheath as it pushes through the soil, so it can come straight up. It doesn't have to form that hook. Okay, so how do plants reproduce asexually? It's quite common, and it can be an advantage to well-adapted individuals. Mostly, you can reproduce a lot of individuals in a short period of time. These are all going to be clones because you're not relying on sexual reproduction by the fusion of egg and sperm. Disadvantages, as always, for asexual reproduction is a lack of genetic variation, which means all of the offspring are going to be genetically identical to the parents. The problem with this is that if you have a disease that affects one of your crops, it's going to affect the entire yield. This is what happened, I think the early 1900s in Ireland. Their main crop was potatoes, and there was a fungus that broke out that was called blight, and millions of people starved to death because it just ruined all of their potato crops. In nature, plants can reproduce sexually through what's called vegetative propagation. This is vegetative reproduction, same thing. This is basically just reproducing with plant parts that aren't egg and sperm, non-reproductive parts like leaves, stems, and roots. So here's just a few examples of vegetative reproduction types. You have in strawberry plants, you've got a runner. This is a horizontal above ground stem that makes leaves and roots at the nodes. Each new plants can grow from each node. So just like where we would think of just a regular branch, it can form an entirely new plant. So that's an example of a runner. And this is a spider plant. We have the rhizome, which is in ferns, horsetails, iris, ginger, and sugarcane. This is very similar to a runner, except it's below ground. So this is, if you dig out the rhizome, that's the rhizome running underground. As you can see here, the iris flower, the rhizome underground. Because normally a stem, you wouldn't consider that a reproductive structure. Something very common is what's called a bulb, a very short underground monocot stem with thick fleshy leaves adapted for storage. Garlic and onion, tulips are examples of this. If you ever want to grow onion, just put an onion bulb in water, and then eventually roots will form and you'll see a green stem come out. If you've ever had onions sitting around in your kitchen for too long, you might actually see a green stem starting to form. Another thing that we eat quite often is a tuber. A tuber is an underground, swollen, fleshy stem specialized for storage. Buds on the tuber can grow into new plants. If you've ever had, again, a very old potato, and you start to see these things start to grow from that, these are buds that can develop into a new plant. Now, we can use the vegetative reproduction to propagate our own plants. So vegetative propagation is our using vegetative structures to produce new plants. Besides using these specialized structures like bulbs, rhizomes, runners, we've also developed methods for propagating from other parts. The most common one is a cutting. A lot of herbs can be propagated this way. If you cut a piece of stem or shoot and you place it in water, roots will form on those cuttings. It's too bad, like it was just last weekend I planted my mint. I could have showed you. I might actually still have the pictures, and I'll send it to you in line, what I mean. So cuttings are pieces of stems and roots that are cut from the plant and used to grow new plants. An example, African violets or any herb that you buy at the market, really. So you cut the stem, and then you can either place that in water to encourage root growth, or just put it directly into soil. Another method is called layering. In some species, roots are going to form on stems where they touch the soil. So people can place branch tips to the soil or cover the base of stems with soil to make them propagate. So layering is the process of causing roots to form on a stem. If you do air layering, you create a wound on the stem. So either you cut a slice into it or you scrape the outer part off, and you place sphagnum moss around the wound. So this is an example. If you kind of tie the stem to the soil and new roots will grow there. Here they've cut part of the branch and then they put moss around it to make it grow new roots. Grafting is joining two or more plant parts to form a single plant. the bud or small stem of one plant is attached to the roots or stems of a second plant. But to be successful, you have to line up the vascular cambium. Otherwise, the xylem and the phloem won't match up, and you won't be able to transport food and water. This allows us to combine different characteristics of cultivars that we like. An example of this, I think they're called pluots. It's a combination of a plum plus an apricot. They're absolutely delicious. I found them here once at Tops at the bottom of Central. And they have different names depending on how much plum and how much apricot there is. And I don't remember the other names. I just remember pluot because that's what they labeled them here. It's more plum than apricot is a pluot. So an example of something else we could do is an apple cultivar that has really good fruit. We can graft that with another cultivar that has disease-resistant roots. So we have a new hybrid that has excellent fruit and disease-resistant roots. The grafting is used to propagate practically all commercial cultivars of fruit and nut trees and many ornamental trees and shrubs. So this is an example of grafting. And the last method that we can use to propagate plants asexually is something called a tissue culture. This is producing new plants from pieces of tissue that are placed on a sterile nutrient medium. Don't really know why you would do tissue cultures unless you're doing genetic manipulations. Maybe with GMOs, you might do that. Okay, so that would be your tissue culture. All right.